Good evening to all. I would like to welcome Arundhati Mehta, Miss Arundhati Mehta, for the session. And Miss Gayatri, are you there?
Yes, I'm here.
Miss Gayatri?
Hello.
Okay,
Hello,
you
am I
can
audible?
start the session. The session is live now. Thank you.
Okay. I now request everyone to mute your mic and switch off the video to avoid any kinds of inconvenience. Thank you. Hello, everyone. A very good evening to one and all gathered here. I'm Gayatri Kanama, and on behalf of Ryan Publishers, it's my greatest pleasure in welcoming you all to today's one-day international webinar on the topic, Edible Vaccine, a Cure for Needle Haters. The participants can post your queries and clarifications in the chat box. And I would also like to inform that the feedback form for today's webinar would be posted in the chat box. It will also be posted in YouTube live chat box. And for your kind information, there are limitations to the number of certificates that can be generated per day. So we appreciate your patience and cooperation in getting the e-certificates for the webinar. Now time to get into the session, and joining with us today is Ms. Arundhati. Welcome, ma'am. Ms. Arundhati is currently doing her PhD in the Department of Biotechnology and specializes in cancer biology. Our resource person has been a junior research fellow and as a research intern. She has done many conferences and seminars. Ms. Arundhati has also received honors and awards, and to mention a few, she has received the Young Scientist Award, BRET Doctoral Research Fellowship, and so on. Our resource person has also shown keen interest in publications and volunteering activities as well. We are extremely delighted to have you amidst us. Thank you very much for joining us today, ma'am. And the session is now yours. Thank you.
Thank you so much, each and everyone present here, and Ryan Publishers, as well as Ms. Gayatri, for helping me out and introducing me to this wonderful webinar. And I'd like to thank all of you to be here. And let's get started. Am I audible?
Yes, ma'am, you are audible.
Yes.
Okay. Thank you so much. So I will be beginning with my talk. Okay. Thank you so much. Are my slides visible?
Yes, ma'am. Yes.
Okay. Thank you so much. Okay, so I'll welcome everyone. So my name is Arundhati Mehta, and I have been already been introduced by our kind hosts. So talking about my topic, that is edible vaccine and a cure for vaccine haters. Basically, I don't like the needles, so that's why I loved this topic, and I really wanted to share with you all guys. Okay. So before proceeding for the vaccines and what is the edible vaccine is, I would like to take a few moments of your time to just revisit our knowledge that how this term came and what actually the vaccine means. So basically the vaccine is just a preparation, biological preparation that is established, and it provides immunity to our body from other harmful ailments and infectious disorders. So the terms vaccine and vaccination was derived from variola vaccinae. That is the first time from where it has been came from, and smallpox that of the cow. It contains an agent that resembles a disease-causing microorganism and is often made from the weakened and killed forms of microbe. That is what we known as attenuated or live forms, or its toxins, or its some surface proteins. Like for COVID vaccine, we are using spike proteins and all. Okay. So generally, vaccines are usually taken care for two types. That is, one is for prophylactic treatments, another is for therapeutic work. Okay. So prophylactic vaccine means is like to prevent the effect of future infection. Like we take all the vaccines during our childhood, that chickenpox vaccine or MMR vaccine or DPT, whatever we take, they are all prophylactic vaccines. They will be guarding our body from any future infections. But there are some therapeutic vaccines also that are used as a therapy. Generally, like in for diabetes, some vaccines have been constructed and it's under the clinical trial, and it's also for the cancer, for preliminary dosage of cancers and preliminary stages of cancer, basically. Some therapeutic vaccines are created and are under the procedure and in the pipeline. So I have tried to make my PPT quite figurative because I think that all this sort of normal information is quite common, everything available on the internet. So I like things to go pictorial. It helps ones to understand, and maybe my PPT can attract you. Okay, so this small picture film, it's already telling us the story of our past, that is from Edward Jenner, 1796, how he used first time the vaccine from smallpox, and it helped the small student, or one week say that a shepherd boy, from help us providing an immunization. So, and that Jenner's work was continued by the Louis Pasteur, right? So you can see here he is just spiking with the knife, taking the sample, then again he is striking the boy's hand, and the immunity is been provided. So, what are the ideal characteristics of a vaccine? So it should be having no pathogenicity or toxicity. So what we can say is that it should be antigenic. Okay, so what the antigenic means that it should be foreign to your body, but it should not provide you with any virulent effect other than its own effect that has been destined. Okay, so pathogenicity or any associated toxicity should be nullified. Okay, low level of side effects. The taken highest vaccines, there are in an year, more than thousands of vaccines are prepared in the laboratory. But many few pass on to the trials, and few of them only came to the market. Why? Because of the side effects and the associated limitations. So the vaccine should have a low level of side effects. No environmental contamination. Wow, we are going go green, and we have to recycle. We are using the 3 R's. So environmental contamination is also because it's a bio waste, whatever we can be saying. Then simple vaccination technique. If a technique is highly complicated, then you will be having, and you will be in need of a different type of manpower to train that people, to have that vaccination technique, to know the protocols. So it should be very simple and obviously economical and cheap. Now we come to the concept of edible vaccine. Edible vaccine, the need or the thing, it goes hand in hand with another contradictory part of our biology, that is genetically modified organisms. Till now, we all that came from the biology background, we all know that still the society has not accepted GMOs, and whatever things that has been accepted is only BT cotton, that is not as an edible form. It has been used for textiles and it's agriculture based. So usually till the time the genetically modified crops or GMOs are not accepted, the plan of edible vaccine is still restricted to that plan. Because what edible vaccine is, it means that we have to introduce a genetic or selected desired genes of encoded antigens. So let's make it very simple to understand. What we have to do is that what a vaccine does in a vial. So we have to make and create exact atmosphere, but in a form of a fruit or a vegetable. So how can we do? We can't just have a syringe, grip a syringe, and you can take out the vial and just fill that thing into the tomato, like my picture is showing. We can't use that, and then after that we can't eat the tomato. because there is a high possibility that vaccine will also trickle down in the form of the juices, right? So if we want to make the route of administration simpler, then we need to modify a regular eating food or any plant product to have that type of encoded antigen inside it. Okay. So as we all know about the golden rice has vitamin A. Okay. So it was planned by the notion that it can help us in controlling the vitamin A deficiency and in iron also. So that type of thing we need. So as you can already see, you can already read the definition I've already written. So it should have its native immunogenic property, but it should not have a virulent property. Okay. Transgenic plants are usually used as vaccine production system. It is a need. Without it, we can't make an edible vaccine. Initially, it was thought to be useful for preventing infectious disease, but currently the plan is also for the autoimmune preventions, birth control, cancer therapy, anything that can be taken in the form of pill can be converted into the form of a food agent. Okay. So very shortly, the thing has been completed and it has come into the research that what happened is that, yes, you can see here, there was a paper in MDPI, that disease prevention and opportunity to expand an edible plant-based vaccine. So the thing people are thinking in the squame and edible vaccine research is in the motion, and they have depicted it in very quite pictorial view that how the edible vaccine research can be take care and how it could be implanted inside our medical systems. Okay, why it is needed? Well, basically, if you will ask me, only one need is that I can do anything if I don't want to prick myself. I just hate it. So still our scientific society, the ethical society, normal people, layman public, everyone wants to know that why then edible vaccine is needed. So what conventional vaccine has provided us, it needed trained personnel, low children compliance. Anyway, no children is ready to take a vaccine that is having this big needle and that is pricking inside their skin. Poor mucosal response. Mucosal response is highly needed because it is one of the main modulatory inflammatory responses. It needs purification, it needs refrigerations, it's fear of contamination, requires syringe, needle that is including to the bio-waste, and obviously it is expensive. Now, what edible vaccine will provide us? It is not needed any trained medical personnel. If you're going to a doctor and you need, say for a vaccine has been created for chickenpox, so what you will go? You will just go to the doctor and the doctor will say, yes. So you need a vaccine for chickenpox? Okay, then you can just grab this apple and the chickenpox vaccine is inside you. So student, obviously children can have that apple in favor of the syringes, right? Obviously, when you will be coming close to my PPTs, into the distal end of my PPTs, you will get to know that how edible vaccine has been undertaken for high mucosal responses. No cold chain required, heat is heat stable and good genetic stability. The people are working on this field. No fear of contamination because the fruits and vegetables are already in the nature is already protecting them. It is a needle free and obviously maybe it will be cheap also. Okay, we have seen the market. Okay, that one, there are two type of apples coming, one with sticker, one with not sticker, one is waxy coating, another with not. So comparatively, it's like one is 50 rupees, other is 55, 60 rupees. Okay, so still it could be cheaper. The history, obviously, we can't move without thanking the people, the person who thought of this idea. So what happened that in 1990s, a Streptococcus mutans surface protein A was expressed in a transgenic tobacco, and then it was given to mice. What was done is that there was a person named Charles Anson. It is a father of medical vaccine. Okay, so in Texas A&M University, he conceived this idea and thought that maybe we can have these plants that is nurturing the humans for this long time could also be a medicine to help and fight our and it's like it's like boosting our immune system. So then this plant came into the existence, and then number of scientists, as you can see in this timeline, Manson et al., Hack et al., McGarvey, Mason, Heen et al., And in different years, that is like till now in 2020, I have said that the people now are walking this lane, and then they have started making that whether we can express the system, whether we can create some sort of an and with the plant or some food or vegetable that can be ingested inside our body. So you can just see, okay, now how it is done, like how this type of vaccine can go inside, like why we say that edible vaccine is possible. So the goal is oral vaccination. We don't want our body to suffer. No more pricking, no more direct inflammatory responses, okay, because what we need is that a vaccine should trigger a mucosal and systemic immunity. A vaccine has to do only that. How it is doing, it is that vaccine's problem. So currently it is doing by going to intraperitoneum, intrav, intramuscular, other types of the route of administration are there. So if we are talking about oral vaccination route, so how it could be taken care. So edible vaccine, when it will be taken up orally, it will undergo mastication process. Yeah, we are eating apple. Apple is degraded in our mouth. Okay, so it's a mastication process. Then majority of plant cell is degraded. That it goes to the intestine, and intestine we know that ultimate digestion in stomach and intestine the goes, and then the lower intestine the absorption level starts. Okay, so in Peyer's patches, they are an enriched source of immunoglobulin A, IgA, that are producing the plasma cells. Okay, so it Peyer's patches that has the ability to produce IgA and have the potential to populate the mucosal tissue. This mucosal tissue is serving as a mucosal immune effector site. This is a biology, it's a core zoologic process. If you want to know, you have to just go a little bit down into the human physiology, basically digestive physiology, and you will get to know that in Peyer's patches, the mucosal immune effect, it is a mucosal immune effector site. Here, the breakdown of edible vaccine near the Peyer's patches, it consists of around 30 to 40 lymphoid nodules on the outer surface of intestine. Okay, so intestine, we know there are villi and follicles and microvilli, and there are many things coming up, right? So in the surface of intestine, it contains follicles. And as we know, that lymphoid system is also running parallel to the blood system. There are lymphoid nodules also. What these follicles do? These follicles act as the site for which the antigen penetrates the intestinal epithelium. Now we know that vaccine, it has an antigen inside it, okay? So what this vaccine is that after the mastication process and official degradation, now the vaccine is inside your body. But to have its response, it has to come in contact with this inflammatory or immune cells, right? So what it is done in this follicles. So this follicle acts as the site, and it will help the antigen to penetrate inside the epithelium. Thereby, it will accumulate antigen within the organized lymphoid structure. The antigen here comes into the contact with M cells. Okay, and this M cells, mucosal cells or mast cells, you can say that these M cells then passes the antigen to the macrophages. And here the story comes to end, because every vaccine has just to come in contact with any MHC or any antigen presenting cells. Macrophages are one of the antigen presenting cells. They are APCs. Okay, so basically, to be very layman, the fact is that this antigen has to come in contact with any of the inflammatory cells or immune system cells. Okay, so this M cells, what it does that it will take antigen from one hand and it gives that antigen to the macrophages from the other. These macrophages will interact with the antigen along with the B cells, and it activates the T cell response. Now, to study how it is coming up, it's the little bit of immunology. So how B cell activation comes, how T cell activation comes, I won't be telling you here. So for that, maybe you need to know and you have to get along to that original official knowledge. But this way, the B cell activates the T cell, and B cell T cell activation comes, and the immune responses will trigger. Okay, it's an overall story. So here I have put it in a very pictorial form that how from the M cells the story goes and goes. Okay. So M cells pass the antigen to macrophages and B cells. You can see in this figure. And in this figure, as you can see, that the antigen is coming from the vaccine. Here, at the from the follicles, it came to M cells. This is the M cell. and is with. From the M cell, it has been grabbed and it has been passed down to the macrophages. Macrophages look like this only, right? Simultaneously, a B cell can also come and B cell can also take up the antigens. Then what macrophages do is that it displays, as per the MHC production, MHC2 production and antigen presentation, this macrophages displays the pieces of antigen to the helper T cells. As we all know, the T helper cells is needed by macrophages. It's a macrophage mediated T cell immunity. Okay, an initial response. And if the B cell is collecting the antigen, it will be having a stimulatory secretional response, and this T cells will stimulate B cell to seek out the antigens to the distant site. Means like the more the antigen will be given, the more the activated B cell response will be triggered. And what B cell will do? T cell has cytotoxic response, but B cell has an antibody response. Okay, so B cell will start producing the antibody. Like if many of the people right now who has taken the COVID-19 vaccine, the doctor has already prescribed you that no, you have to not do this, you have to do this. For 1 week, you have to be very much calm, pose. Why? Because your body is preparing the antibody, so it doesn't need more drugs. Or taking any vaccination, while swine flu vaccination, chickenpox vaccination, MMR vaccination, the doctors always say that take rest, don't overexert your body because your body is into the process of forming of an antibody. Okay? So activated B cell makes and release the antibody and be able to antigen. This is an initial response. That is when vaccine done. But it is not needed. Now that currently you have taken an antigen and you are just going out and there will be a problem and some of the known, foreign antigen, okay, it will be attacking your body. So taking the COVID example is like if you have taken the COVID-19 vaccine, so your antibody has been created inside the body. Okay, so now if you will be going out, okay, so after you will be going out, there are many chances that actual COVID-19 virus will go inside your body and strike and reaction. But the COVID is not knowing that your body is already having an inflammatory response memorized inside, right? So the memory response is very much needed because it is the only thing that helps to, which is triggered. Okay, then when that actual reaction or actual war takes, it's a memory. It's like there's already the soldiers are prepared for the war. Okay, so memory response, the T helper cells brought the cytotoxic T cells to attack the infected cells, and this memory helper cells. So the virus is arriving, okay, you can see here, and the same thing, like in the previous figure where I have shown that triangles, now here actually this virus is coming. The virus was not knowing that our body is already knowing what to do. He is thinking that the body, it will be coming inside the body and it will trigger its reaction. But no, our body already knew that if something like this comes, what to do. So what they will do is that automatically the macrophage will come, engulf it, present it to the T cell. The T helper cell will, so in the no course of time, will share an immune response that is having a memory mediated response, and it will automatically tell T cells, the cytotoxic cell, that it is the infected thing, kill it, burn it, whatever you want to do, just do it, just eliminate it outside our body. Okay, simultaneously the B cell will trigger, and without wasting a single moment of time, the memory B cell will tell that no, not needed, it's not our guest, let them go, they're just not needed, just squeeze out the antibodies and neutralize the invader. Okay, so the things are done. So the thing that has taken and it has been incubated inside the body, when you have actually taken the vaccine, it took much time. You have taken right now the vaccine, the response has been triggered for 10 days, 20 days, 30 days. Okay, so it is an incubation period. But when you'll be actually infected by the virus, you don't have that much of time because virus currently continues to do the work and it infects the body, throws out n number of responses, and you're hospitalized. And as we know that this pandemic situation is there, it is so risky that we can't even waste a single moment of time. So the virus don't know about it. So to limit down that procedure, that in place the virus think that now I have to just do this and do that in the body. Your body already know that you have to do nothing doing. You're just going out of the body in the killed form. Okay. Now, what is the second thing? As I have already said, that not only for the prophylactic the vaccines are by design, they are also designed for some therapeutic effect. So one such demand was also for fighting it off from the autoimmunity. What autoimmunity is that? If your body is starting treating started treating your own proteins, your own response as a foreign. It happens in many autoimmunity, where one such type is type 1 diabetes, where your own proteins that has been secreted from the pancreatic beta cells, it becomes foreign to your body, and your own body and your body starts working against that antigen. Okay, so it's like autoantigenic effect. So you can see that before the treatment, how the person has been striving with the type 1 diabetes. Hello? Is there any problem?
No, ma'am, you can proceed.
Okay. So as you can see, this the before the treatment, the person is having a real problem, and so many type of inflammatory responses and the suppression and the triggers that are undertaking in the normal type 1 diabetes. But if it undertakes the treatment, what it has with normal vaccine treatment, so the body memorizes, the body gets to know that the autoantigens that has been created outside the body, inside our body. See, so let me just explain you this because it's a little bit tricky. What we are thinking right now is that, so what happens is like this is our beta cells that is under the attack in pancreas, and it is having some damaged area, and many of our inflammatory cells like natural killer, macrophages and B cells and T cells, everyone is coming and attacking it because these cells are thinking that, oh, this is the foreign material. But if the vaccine has been created, then what it will do is that it will just have this protein, is like the vaccine will just again make these all cells understand that no, this is not foreign. And what it will do is that it will suppress the T cells. As we know, the T cytotoxic cells is like all the cytotoxic effect needs helper cells. Without the TH cells, the cytotoxic cell can't activate. So this type of vaccine that has been prepared or under the process of preparation, what they does is like that they suppresses our inflammatory cells. Okay, so thus or indirectly they are preserving the beta cells. Now there are some targeted treatment also coming up with vaccines that are making cells understand that no, it's not foreign, and still the autoimmunity is under the control. But this thing already, and it requires more research and more paradigm shift, actually, I will say. So what is the developing procedure? Till now we have understood what is the need, how it came, how it works, and now how can we do it? How can we prepare it? So the developing procedure is like it can be made by two ways. The ways are simple. All the things is like edible word is new because the system is not that glass vial. It's just a common fruit. That's why it is stated as edible vaccine. The development procedure is quite similar. Okay. So what is the first stage, or what we can say first way is that if we take the entire structure gene of the antigen. Okay. So coming to my right side of the figure, you can see there I have sketched out in a black and white form. Here it has been written on the top that antigen from the pathogenic microorganism has been taken. Okay, so it has been taken, and now it can go to the two ways. One is using that entire gene, the structure gene, and undertaking it in the transformation vector. So transformation procedure is undertaken. It's a molecular biology technique where we introduce the foreign gene inside our known expression system. Usually our expression system is bacteria, E. coli, or virus. It depends on, but usually it has been taken E. coli and some known plasmid vectors are undertaken. So the transformation vector, it has been undertaken, and then it has been carried forward for the agrobacterium mediated genetic transformation. Many of the people who are from biology background, they already know that agrobacterium mediated transformation or agrobacterium tumorifications, TI plasmid and all. So if you don't know, kindly tell so I can help you know first thing that after, because this procedure is similar. It's like it has nothing to do. Once I will be just saying that agrobacterium mediated transformation is undertaken. Okay. So from via agrobacterium mediated transformation, it has been stabilized into the nuclear gene. Okay. And for example, here it has been taken a potato plant. Okay. So here as a potato plant has been taken, so that structure gene which was expressed in our system, in our MCS system, Then it has been undertaken for the agrobacterium mediated transformation, and it has been transfected inside the plants. And once this type of plant starts to bear the fruit or the vegetable tuber or anything like that, but then it has been checked that these type of plant or the fruit of that plant is having that foreign gene inside or not. And once the gene is been done by a normal expression pattern study and other, you will get to know that yes, it is ready for the immunogenicity study. What is the other way? The other way is that when we are talking about the antigen could be a small gene, and it can also be a part of it. Okay, so what I wanted to say is that taking an example again of a COVID, here we always say that the spike proteins are used for as an antigen, and first initially when the vaccine plan was created, everyone has thought of creating a vaccine taking as an antigen of that spike protein. Obviously vaccines, these viruses are rapidly multiplying, and they are changing and having a great high retention rate of mutations, so it is not possible. But still the plan was there of having the spike protein as one of the antigen. So and every antigen has one expressing site. Every antigen, kindly note this word, because every antigen has an expression site, and that is known as epitope. What are epitope? Epitopes are actually that space, that that regions which goes and binds to antibody. Okay, so the antibody is one thing, antigen is another thing. We know that they both work in a lock and key mechanism. Antibody binds to the antigen, but there should be some surface, right, from how they will be binding. So antigen has an epitope and antibody has a paratope. This epitopes goes and binds to the paratope. Okay, so either you can take a whole antigen and work with them, that is called whole structural gene, or either you can just take that epitope site, okay, the DNA fragment of that epitope, which is that identification or characteristic feature of that, and you can use it to create a vaccine. So the second way is undertaken as a epitope. So epitope fusions are created by a chimeric gene. Like you can take here the plantation, say you can say the tobacco is in there, like tobacco mosaic virus, it is already used there, and identification and integration of that fragment, and then construction of a gene by you fusing the core proteins, and then again infection to the plant of that modified vaccine, and then isolation of the chimeric viral protein. But these developing procedures are lengthy. Obviously, they are quite regulated. They should be regulated, because if you miss anything, there are chances that you may be thinking that this fruit or vegetable contains the vaccine, but maybe due to some mutation or some missing in that fluke or in the fluke, the thing is not that great. So you have to keep a very concrete check. And yes, it's one of the disadvantages that I will be dealing with the edible vaccine. There are some factors that are affecting the efficacy of edible vaccines. The antigen selections, efficacy of model system, they are very, this 7 points are really very much, the one has to undertake into the notification, because you have to check it. So which will be the part that would be you taking as an antigen? What is the efficacy of your model system is like, if you know that your vaccine is very much, it's not heat resistant, like you can't make any of the genetic transformation in that which is heat resistant. So you can't put that gene vaccine into the potato, because a person cannot eat raw potato. It will be boiling it, chopping it, frying it, doing anything with it. Okay, so you can just go for it. So you have to be very much in a plan, having a good blueprint that how you would be making it. The choice of the plant species may be in one species it is fine, other species there are some complications. As we can see, there are a number of species of wheat, rice, mangoes, like many things. So one has to screen out very particularly. Delivery and dosing issues, yes, because obviously if you will be asking for the expression pattern, the expression should be regulated. The promoter, the enhancer, or the blocker, everything should be regulated system, because once it goes inside the body, the role is finished, the film is gone. You just don't have any time for any teaser or it. There will be direct climax. There will be nothing. So delivery and dosing issues are really needed. Obviously, safety issues, because it is concerning a human health. You can't just compromise with the safety issues. Public perceptions, attitudes, genetic modification. Public perceptions are still not fine. They are still not agreeing to the genetically modified organisms. So one has to make it very much comfortable, and people has to be very consent to it. One can't force them to. And obviously quality control and license. Methods of transformation of DNA into the plant and gene. So I will have to rush because it's already 5:36. Okay. So methods of transformation DNA. So there I had stated that there is one method known as agrobacterium tumefaciens method. So what is that method I will be dealing here? So it is a vector mediated nuclear transformation. It is in general form, it is known as vector mediated method. So here in a very normal form, what we have to do is that we have to cut a leaf. Okay, so a leaf has been cut, and what we have to do, then a Petri plate containing that bacterial cell. That which bacterial cell? Are plasmid. Okay, that plasmid also has to be 2 to 3 things. One that bacterial cell that is having an antigenic gene, the other it has a plasmid, and it should also have antibiotic resistant gene. Why it is needed? Because if you recall your molecular biology protocols, you need some good cloning sites, you need multiple cloning site, and obviously you need a selectable marker. You will never get to know whether you're actually the gene has been incorporated inside your frame or not. So there has to be a good selectable marker. So antibiotic resistant gene work as a selectable marker. So we just expose the leaf to the bacterial function and allow the bacteria to deliver the cells, and by normal conjugation procedure, plasmid and plant cells, here you can see the DNA has been transported inside the plant cell from the DNA. The gene transfer is undertaken. Dead cells and again everything, the media, the cells, everything are put under the control. And then this leaf is being checked that whether the transformation is been undertaken or not. Okay, so how it has been done, as I have already said, that there will be the selectable marker, and that selectable marker gene expression should be coinciding with the gene expression. So that gene we are making, if we are taking antibiotic resistance as a gene marker like chloramphenicol or ampicillin, so we will be having an ampicillin solution. We will be going and putting that plant tissue into the plant cell into that antibiotic solution. We will be waiting that which type of cells thrives, which type of cells lives. It's many things like making an hybrid cells. Okay, you if you have known about that thing also. So it's a very normal, it's screening actually. Okay, so it's a normal. Then we will allow, as we will get to know whatever the cells live that has our gene of interest incorporated. So I will let them grow into the form of callus, and we know that from the plant tissue culture that callus are responsible and have that integrity to create it to the new cell. So this callus will be sprouted, giving out shoots and roots. Then we will put it in the soil, and then the plant research comes into the existence, and yes, then we will be waiting for the vaccine potato, for example, or any transgenic potato or transgenic tomato or anything. So it is agrobacterium tumefaciens mediated vector system. Okay, yeah, bacterial cells, obviously, agrobacterium tumefaciens is one of the really known cloning system. It has been well advised to use this. Vectorless. Well, obviously, there is some people are not interested to use the vector system. So what they do is that they use some vectorless or direct gene transfer techniques. So what they have, one is the microprojectile bombardment. Here you can see in this figure, microprojectile bombardment. Here in the very short I will be telling you is that your gene or your gene of interest has been coated on gold or tungsten particle, and they are then loaded into the gun. Okay, so gene guns. And then they are bombarded inside the plant cell. So the plant cell directly undertakes the gene of interest. Okay, that the DNA is going inside the cell. All these things are just done so that our DNA that is residing outside goes inside the plant cell. Okay. The next process is the electroporation method. Electroporation is creating or giving a pulse of high voltage. So we all know that cell membrane has some charges. They are differentially present inside and outside the membranes. So before this pulse, we can see that plus minus, they are equal. Then we need to introduce the gene. So what we'll be doing that, providing an electromagnetic field. The electric field induces the voltage across the cell membrane, and we will see that all negative comes one side, positive comes one side. So all this, the green wavy structures are DNA. So creating that electromagnetic field will help us providing a loop, a tunnel, a canal, whatever we can say, and our foreign DNA goes inside the cell. And we will just give a pulse and the pulse stops. Once the pulse stops, the cell again heals themselves and our DNA is inside. Our work is done. What is the third? The third is micro injection. Here you can see it very coolly. Micro injection, I have thought of giving a good animation. Here you have to poke inside the cell. Okay, poke and just transfer your sense and gene of interest inside the needle. The next is the chemical mediated transfer. The chemical mediated transfer using a pegylated capsules, dextran sulfate, calcium phosphate, or anything that like CaCl2, how we can make the competent cells the similar sort. It is just a procedure how we are making a competent cell from the transfection. Next is liposuction. Liposuction is using the liposome. It's like we have to encapsulate our DNA into the liposome. Then liposome is a DNA lipid complex, and it will be easily undertaken by the plant cell by the process of endocytosis, because liposomes outer structures are very much common with the plant cell. Okay, outer plant cell. So by the process of endocytosis, people tend to use it very rarely, because there are also some fluctuating chances of using the liposuction. Obviously, the next is conjugation. It's a very normal bacterial conjugation, performance and preparation of relaxosomes and transferosomes. Now, what are the generally plants that are being undertaken currently? So tobacco is used, potato, banana, there are list of plants, and people are thinking, the researchers are screening out this plant as per their need, as per their choice, their area, their route of administration. So it differs. And each and every plant has its own pros and cons. It's like potato, it can't be cooked, but it can be ingested. Many people like potato. Simultaneously, for the muskmelon can be eaten, but many people don't like. Personally, I don't like muskmelon. So the type of plants are just, it's with the researcher, it's with the checker, it's with the planner who is using and how the things are undertaken. Okay. The comparison of the plants, as I have said, there are advantages and disadvantages associated commonly with the plants. So tobacco, it's a good model for evaluating recombinant protein. It's low cost, easy purification, but obviously tobacco has a toxic compound. And obviously you can't say and you can't give anyone that this is tobacco, but it's a vaccine and you can take it. Obviously, it's the consent issues also, right? Simultaneously, potato is very much dominated in clinical trials. It's easily manipulated, transported, but it needs cooking. So antigens can denature. And obviously, immunogenicity is also compromised. Banana, no need of cooking, proteins are not destroyed. But if you are using trees of banana, or we can say banana is a plant, actually it's not a tree. So if you're using a banana plant, so obviously it will take 2 to 3 years of mature. But obviously the plant tissue culture technique is helping out here. Bonsais is also helping out here. So it will take 2 to 3 years to mature, and transform trees are taken back in 12 months. And obviously, if you're talking fruit, it's easy to eat, but there is a problem with ripening. Okay. And for vegetables, they are not easy to eat, but they have a long longevity. So simultaneously, you have to know that which thing you want to take, which plant model organism you want to take. There are other examples like tomato, rice, lettuce, soybean, alfalfa, muskmelon, others, and the list goes on. Obviously, this data, everything I try to incorporate it some papers. So if anyone of you is interested to learn, to write anything on this, you can use this website or this research papers and these authors, because these authors really had brains and they had done really good jobs working on these things. Now, we're coming to the end of my presentations, and so this is the advantages. Some we have already dealt. The edible vaccine, it means administration reduces need of medical personnel. It is not needing any storage. It's a normal storage, possible productions, easy separation, antigen protection, enhanced compliance, because you can easily give an apple to a person and they will happily eat it. Effective prevention and pathogenic contamination from animal cells, which is economical, and simultaneously, as the point, it has a good disadvantages in a big list, where the one most big disadvantage is that acceptance to the genetically modified plants. Like people usually don't take it. There is not good work right now that proves that yes, one can take genetically modified. So the researchers are still working, promoting the research and accepting. And obviously, Now the norms have been created, the norms are modified, and even the ethical committee is also in one of the panels selecting that whether this type of plant should be undertaken for genetical modification or not. So development, immunotolerance, consistency, dosage. Dosage is a main problem, that maybe an apple looks red, very much red, other is also looking very much red, but there may be some titer problem. Vaccine one in titer is different, other is true. So there is a need that regulation is needed. There is no checkpoint. Selection of the best plant is difficult. There is till now not one of the best plant that will be taken for use. And there are certain foods like potato, obviously it can't be eaten raw. So obviously in infants for which we use that vaccine chart, it's not possible because infant can't eat. Okay. Clinical trials. So talking about some real things that how this thing are proceeding right now. So there are some clinical trials on transgenic potato, that is with CTD genes. Vibrio cholera was shown to be effective in mice. Eating one potato a week for a month with periodic booster was said to provide immunity. For Norwalk virus, transgenic potato was used where 19, that is 19 out of people fed with transgenic potato expressing antigen shows seroconversions. There is a problem. Hepatitis B, parental BLPs, the transgenic potato is used. For measles, transgenic rice, reduce and baby food against the measles are being developed. The mice is fed with the tobacco expressing MVH, attain antibody titer, enterotoxigenic E. coli for gastroenteritis. Okay, so transgenic potatoes are used. So basically, potato is one of the main things that we are already using. And then the applications. As I've already said, that has two applications, prophylactic and theoretic. So infectious disease, there is a long list of infectious disease. The first are the care of autoimmune, type 1, multiple sclerosis, and cancer therapy. For colon cancer and cervical cancer, the people are thinking of using edible vaccine, and they are planning to do the research. Here, one of the research article I came across and I thought to put it in, that is edible vaccine for the COVID-19. A person named Syed Sartaj Saurabh, he has given this article in the clinical experiments of vaccine residency, and in edible vaccine development for coronavirus 2019, the concept. Here, he has very much said that maybe the spike protein can be taken. It is just a concept, obviously, but the people are thinking for COVID-19 to curb out and to be in the form of the edible vaccine. This is in the paper. Again, there's a new list. It's just a 2020 list, and here you can see many of the viruses have already in the delivery in the phase, the different phases. One is in the market also, hepatitis B, tobacco, but don't know that the doctors are prescribing it or not. Okay, so here, so there are some of my references which I've usually taken back. Maximum my images are from the Google, so obviously I want to thank them. Thank the Google. Yeah, I just want to take a small time to just introduce myself, that this is my university, GGU, Vishwavidyalaya. That's my department, and here these are my teammates and my supervisor and my seniors and all. So with this, I just put an end to my talk by saying that yes, let food be thy medicine and medicine be thy cure. Thank you so much to be my patient listener, and I will welcome any questions if there are. Thank you so much.
Thank you, ma'am. Thank you for the informative session.
Thank you.
Dear participants, you can post your questions in the chat box, or you can unmute your mic and ask your question. Ma'am, there's a question from Surya posted in the chat box.
Yeah.
I'll read it for you. Of the vaccines discovered now, to your perspective, which one has the potential to fight against corona?
Okay. So when we are talking about having a fight against the corona, it's not that simple, because till now, it's a salute to all the researchers, the Serum Institute and everyone, that they have already created a vaccine, Covishield, AstraZeneca, etc. But even they are knowing that 2 doses are needed, and still the people are also having this sort of a corona infections. So if you're talking about the edible vaccine for the corona, obviously the first option will be gone to transgenic potato to use it. And for the vaccines that have been discovered, and if you want to incorporate these vaccines into the edible, into the plants, so it's not that easy. As I've already said, that we have a long journey, long war to take, because first we have to make the people understand that not every genetically modified organism, that is a plant, GMPs, are not bad for health, because our history has that type of records with that BT sugarcane and BT brinjal and all. So people first need to know that maybe the thing is safe, and after that we have to plan. And it's not a one people job. It will need a panel of people, virologists, microbiologists, and plant tissue culturist, and the market researcher, the ethic commission, everyone. So it will not need my perspective, because basically I'm not from virology background, I'm from cancer biology background. So I like this topic. I love to write on COVID-19 and all. So basically I thought of using edible vaccine as a topic. But thank you so much for your question. It's really great. Anything next? What's the difference between Covishield and Covaxin? I just understand. It's like you're asking me to know or anything. Excuse me. Ramesh Yamgar.
Ma'am, I think they've asked that as a question to you.
No, obviously I know, but what he wants to know is like whether he wants to know the difference between the Covaxin and Covishield vaccine difference, or he just wants to know that how can I differentiate between them? It's like for the efficiency he's asking, or what the regular difference he's asking?
Regular as well as the efficiency, ma'am.
Oh, okay. So there are actually two. Basically, there is a good difference between that. One is in the inactivated form, and the other, you can say that it's an live attenuated form. Okay. So let me make it very clear. Covaxin is an inactivated form of vaccine. Okay. And the Covishield, what we are talking about, is a viral vector protein, which is non-replicating. It's a part of a virus that is not replicating. It's stagnant. It's a housekeeping. Okay, so there is a basic this difference. There is other difference in the dosage. Obviously, the one needs 2 dosage, but the difference is in around 1 to 14, 1 to 20th day. The other has been changed right now to around 30 to 60 days. The dosing, the one needs, if you're taking a day one dosing, the other, if you're taking Covaxin, then you have to take the other dose in 20th or 30th day. But if you're taking Covishield, then you have the duration has been changed to around 30, 35 or 40, 43rd day. So I usually know only 2 differences. Maybe you will get more if you will be having that you may undertake the new research. Basically, there are 2. And obviously the developers are different.
Thank you, ma'am.
Okay. How much it is necessary for all of us to have a vaccination, ma'am? Can you please explain it in nutshell? If you are asking me this question to rate it in the scale, then I will be scaling it as 9 out of 10. Because one, I always leave, being a scientific student, we always undertake the errors in our hand, because some people are allergic, so we can't force that people. But to me, I personally think that yes, the vaccination is needed for us. Why? Because I can say it in a different form also, that if the vaccination would have not been needed, then the mother nature would have not provided us anything like innate immunity. Okay? So innate immunity is been inside our body. It has been passed on from a mother to a son or a daughter. So vaccination is needed, irrespective a person is having a strong defense that yes, I'm having some sort of an allergic reactions, or my body is not accepting. So I personally think that yes, it is needed. It is very much needed. What is that? I have one doubt. Only one booster dose is sufficient for COVID? No. Basically, that's still a theory of contradiction, I will say, because what we don't know basically how it has been prepared and what the researchers has planned. But when we say that there is a need or there is a dosing, then because the scientist or the researchers who has created it, they know that taking this one titer will not give that ample of reaction to your body or that memory response. So when we are talking about a dosage, that you have to take a 2 dosage at this interval of time, that is in your body, it should be a successive release, and it should have that much long, in one word, what I will be saying is like that counter attack. Okay, so your body needs to know that yes, one antigen is going inside your body. And for creating that memory response, maybe it will need that type of time. So, as if one is taking a Covaxin for 24 days, 30 days, it will need. And for 30 days, your body is continuously told that yes, the body is having some sort of reactions. And if this thing comes, so it's like basically the weak student pattern. I will be saying that. Okay, so a student who is weak, that the teacher has to work on, and that student has to compulsorily study. And they say that yes, I have to read this line 2 times, 10 times, 5 times. So our body needs to learn that line for around 20 days. So, but when we are asked about other thing, that is MMR's vaccines are undertaken, or that chickenpox vaccine is taken, they're a very sharp student. So when we'll be just injecting one time, the body knows that no, if this student is come, get out. Okay, so this story is like that. So I will say that booster dosage are very much in compliance with the clinicians and the persons who are creating it. Anything else? Maybe I have made many things clear. Maybe some people could not be in lane with me. So I tried to make it clear. Obviously, I accept that I'm not purely from the virology background. I have my bachelor's in microbial technology. So I know I am from biotech background, so I know a little bit, but many things you guys being from microbiology, the other persons, they know very well about from me. So I'll be happy to learn from you. If you not compliance with my idea, I'll be happy if you accept my idea. And if anything goes well, I'm really sorry for that also. So anything else, I'm happy to take.
Thank you, ma'am. Thank you so much for answering the questions. Dear participants, do we have more questions? Ma'am, I think the question session is over. Thank you, ma'am. Thank you very much for your time. The session was very informative and it was very useful for the current scenario. Thank you so much for your time. And I would like to extend my thanks to all the participants for your patient listening as well. And the link for the feedback form has been posted in the chat box. Please do fill it out. Thank you. Thank you, one and all.
Thank you so much, everyone, for your patience listening. And thank you so much, Giant Publishers, for providing me with this wonderful opportunity.
Thank you, Ms. Arundhati Mehta, for this wonderful session. Thank you so much.
Okay.